So good afternoon, everybody. My name is Allison Littrell, and with the support of Dr. Drace in his lab, I study the effect that Efflux pump inhibitors have on the bacterial growth in the presence of lead nitrate. So if any of you have ever traveled outside of the state, going to the beach or Gatlinburg or just going on a trip, and then came back via the interstate, you most likely passed this sign that says, "Welcome to Alabama the Beautiful." In fact, Alabama is ranked #1 this side of the Mississippi River whenever it comes to statewide biodiversity. However, on the flip side of that, we're also ranked #10 in the nation whenever it comes to statewide pollution. The Birmingham-Hoover area is actually ranked the 8th highest city whenever it comes to air pollution in the United States. So with us being so highly ranked with the biodiversity, but also with pollution, the effects of heavy metal pollution is something very important that we need to study. So what causes heavy metal pollution? Just to clear the air a little bit, a heavy metal is any element that has metallic characteristics and toxic effects at low concentrations. So in this periodic table, the elements that are red, orange, and yellow, they're actually your main heavy metals. So the cause of heavy metal pollution can range from industrial waste, wastewater from certain factories, sewage sludge, but then also the smelting of ores like one would have seen in Slush Furnace whenever it was up and functioning back in the day. So there are many effects, again, as I said, for heavy metal pollution, some of the more minor being just having trouble breathing or having coughing fits, to more severe being lung cancer or even neurological diseases. So the high levels of lead have effects on prokaryotic cells and eukaryotic cells alike. Some of these effects being protein dysfunction, the production of ROS or reactive oxidative species and antioxidant depletion, the impaired membrane function, which then in turn affects the nutrient uptake of the cell, and then also genotoxicity. All of these either affect the DNA of the cell, the protein, or the cell membrane of the cell itself. So over the years, certain strains of bacteria have evolved to become resistant to heavy metals, and that just means that whenever they are in the presence of heavy metals in their environment, they are able to persist and to continue growing. This is really important to study because whenever you find a bacteria that is resistant to heavy metals, they could use the same mechanism to be resistant to antibiotics, and that's really important because if this bacteria is harmful to your body and it gets in your body and you can't treat it with antibiotics, then you're going to have to change your whole medical regime to be able to fight off that certain bacteria. So there are different mechanisms of lead resistant, one being lead sequestration, where the lead just sequesters and places in and out of the cell surface absorption, lead precipitation, and efflux pumps. And efflux pumps is actually the type of mechanism that I studied in my research. So I do have an animation that I made for efflux pumps. So this is your Mr. Bacteria, and it does have the efflux pump present. So this certain strain has been grown in an environment that is very heavy in lead concentration. So as it has grown through the years, it's started to accumulate lead. And the way that it's going to get this lead out of the cell is by using the efflux pump. So the efflux pump is going to be activated by a molecule called ATP, and that will open up the efflux pump and allow a lead molecule to go inside of the pump. From there, the ATP is hydrolyzed into ADP, and then the lead is allowed to go out of the cell. So this continues until all of the lead is either pumped out of the cell or the lead runs out of ATP or its energy source. So my research in specifics tried to determine what would happen if we knocked this efflux out and made it non-functional. So there have been past studies here at BSC that have been like this. In 2017, Brittany Files actually did the same experiment except for with mercury. She looked at the inhibition of efflux pumps using this inhibitor. That's the structure of it. It's just phenylalanine arginine beta-ethylamide, or just PA-beta-N. And the way that she quantified her results were through the minimum inhibitory concentration and the minimum lethal concentration, or the MIC and the MLC. So to kind of explain what the MIC and the MLC is and how you quantify that, you have a sample of media that it has ranging concentrations from low to high of either heavy metals or antibiotics or whatever you're testing, and you place a sample of bacteria in each of those conical tubes and let that grow overnight and see if there's growth. And then the lowest concentration where you did not see growth is the minimum inhibitory concentration. From there, the samples that did not grow, you transfer those into a new media and allow those to grow overnight. And the lowest concentration that did not grow is the minimum lethal concentration, because the cells were actually killed off rather than just inhibited when growing. So this is actually a graph from Brittany Fowles' paper. The x-axis is the mercury concentration and the y-axis is the absorbance at 600 nanometers. The absorbance here in the bacteria growth has a direct relationship, so the higher the absorbance, the more bacteria have grown. With the red line is when the Efflux pump was functioning, and the blue line is when the Efflux pump was non-functioning. So whenever we're looking at a graph like this, we're looking at a change in pattern, not necessarily a change in value, because there is different bacteria strains grow at different rates. So with the blue line, you can see that the big drop-off is from 100 to 125, so that means that the growth was inhibited at the concentration of 125. The red line, whenever the Efflux pump was functioning, it did not drop off until it hit 225. So with this, the minimum inhibitory concentration with the Efflux pump was non-functioning actually decreased. And in this sample specifically, she also found that it decreased with the minimum lethal concentration as well. So what am I doing? I'm trying to answer the question, how much of an influence the Efflux pump have on the resistance to lead nitrate in certain strains of bacteria. If we hypothesized that if the Efflux pumps were non-functional, then there would be a lower resistance to heavy metals. Therefore, the minimum inhibitory concentration and the minimum lethal concentration will both decrease. So what did I do? The first thing that I did was recategorize and regrew bacteria that were from the Bio 110 Cell Molec lab from last semester. They had to report whether or not these samples were lead resistant, so I just picked out the ones that they said were lead resistant and then regrew them, so I had fresh samples to work with. And then I confirmed that resistance by growing them on a lead-based plate. 2 of them ended up not being lead resistant. I guess there was just some miscommunication there. And from there, I actually just worked with 8 samples just because of time constraints. It was getting kind of late in the semester whenever we got to this stage. So each sample got a 48 well plate, just like this one. Rows A through C had the Efflux pump inhibitor, so it was non-functional. And rows D through F, the Efflux pump was functioning perfectly. In column 1, this served as our negative control, so it was just 5x PYT80, which is the media that we grow bacteria in. Column 2 was our positive control that had the PYT80 along with the bacteria. Columns 3 through 8 were our experimentals, with 8 being the highest concentration of lead, being 0.25 molar. And then we did a serial dilution all the way down to column 3, with the concentration being 0.0078125 molar. So from that, we let the cultures grow overnight. We had a 24-hour incubation time, and then we read the plates at 600 nanometers. And from there, we made a graph and determined the MIC and the MLC of each sample. So just to restate my hypothesis, we said that when the Efflux pump was non-functional, the MIC and the MLC will both decrease. This is a representative of all of my samples. I did 8, but I'm just going to show you one for right now. This is strain ACH3S2. The x-axis is lead nitrate concentration. The y-axis is the absorbance. The blue line is whenever the Efflux pump was non-functional, and the red line is whenever it was functioning fine. So first off, you can see we're looking for a change in pattern. There's not really a change in pattern. The lines are pretty much parallel all the way through. So that means that the Efflux pump inhibitor didn't really have an effect. They both drop off. You can see right here at 0.0625, so both were inhibited at the same concentration. With the MLC, we did kind of see a little bit different trend. Well, really we didn't see a trend at all, but with this one we actually saw an increase with the inhibitor, but you'll see in a second that didn't really pan for the rest of the samples. So here are all 8 of my graphs. We're not really going to talk about each one of them, but you can kind of see overall that all of the graphs have that same parallel line where there aren't really any big gaps in between. So what are the trends? As I said, with the MIC, we saw no difference between any of the samples. They all dropped off at the same time. Every single one of them had, there wasn't any change with the functioning and the non-functioning Efflux pump. However, we did see a little bit of difference with the MLC. We had 3 samples that actually increased. We had 3 samples that decreased, and then 2 samples that didn't have any change at all. So from this, we couldn't really have a steady trend that we could conclude. So what do the results mean? For all 8 of my samples, 0 were significantly different. So this means that I rejected my hypothesis that efflux pump inhibitors have an effect on the growth in these tested concentrations. There is a possibility that the nitrate had an unknown effect. So you can see here in this graph, it's the same graph that I showed you earlier, that these first 2, that's where the lead nitrate was at 0, but then there's an increase of growth whenever the lead nitrate is at its lowest concentration. And because nitrate is just a compound that kind of promotes growth, we think that since the lead was in such a small concentration that the nitrate kind of made it grow a little bit better. So to kind of help with that in future studies, I think that we could use a nitrate-based media in the control to kind of test for that. So where do we go from here? I do think that we need to check for antibiotic resistance because this is kind of the whole point of the efflux pumps. If certain strains have lead resistance through efflux pumps, they could also use that with antibiotics. So I think it would be really important to really focus on that. Since the efflux pumps didn't necessarily work in my study, I think that it would be important to look at other mechanisms of lead resistance, maybe lead sequestration or even membrane absorption, and also compare the efficiency of the efflux pumps with multiple heavy metals. So in 2017, with Brittany Files, it seemed that the efflux pumps pumped out mercury just fine, but with me, the lead, it seemed to not really have an effect. So maybe designing an experiment where you could compare one strain or a couple of strains of bacteria with different types of heavy metals to see if the efflux pump had an affinity for a certain heavy metal. So I would just like to thank Dr. Kevin Drace for letting me work in his lab, supporting me throughout the semester, Dr. Duncan and my Bio 475 classmates for helping me develop this talk and critiquing me throughout the semester, the Birmingham Southern College Biology Department for everything they do for us students, and then the fall 2018 Bio Winton students for allowing me to use their samples. So here's my work cited. Thank you, and any questions?
We have time for questions.
Are there different types of efflux pumps that maybe you could like specifically look individually at? Like maybe you didn't see that trend, like it was like a general trend, but you might see one that you specifically look at specifically.
Yeah, the efflux pump inhibitor that we used was just a generic efflux pump inhibitor, so I'm assuming that whenever we put it in there, it just knocked out all of the efflux pumps, but that'd be something that we could look at in the future.
Did you do any experiments with the same strain that Brittany was using?
No, ma'am. We just used samples from the Bio 110.
What year did you say Brittany's was?
2017.
So the trains were dead.
Yeah, probably.
So of the other mechanisms of resistance you mentioned, which one would you choose next if you could continue to study? Which one would you study next and why?
I think the sequestration would be something really, not necessarily easy to study, but I think it would be interesting because you can kind of, how Joseph, with his study, found the agrosomes, that would be kind of the same mechanism. You could fluorescently tag certain lead and then kind of put them in the bacteria and then let them grow over whatever and see if there are any aggregates in or around the cell.
So I may have missed this, but you said you verified the lead resistance that we used. Did you verify the antibiotic resistance, or were any of them antibiotic resistant?
We tested one strain with antibiotic resistance because we had a little trouble with the efflux pump inhibitor at the first go around because we were using an old efflux inhibitor from last semester or a couple years ago. So we ordered a new one, we got it in, and we were just checking for resistant, just making sure that it worked. And the strain that we actually tested was not antibiotic resistant. It killed everything. So it's not for sure, but yeah, that's the only one that I tested with antibiotics.
Yeah, well, thank you very much.